So the Open Innovation Portal that AstraZeneca operate is really its collaboration tool to enable the broader scientific community to access the infrastructure, technologies, compounds, cell lines, for example, that we have within the AstraZeneca family. So mass spectrometry is a very interesting technology, and we're very interested in using it for delivering high throughput screening. Its major benefit is really the fact that it's label free, so it's intrinsically fairly low cost to deliver. The problem that it has though is that most of the traditional technologies require some kind of chromatography. Chromatography is generally fairly slow, so therefore it's not really compatible with high throughput screening. So we've used acoustic technology within our compound management area probably for the last 10 years, and we've intrinsically used it just to move compounds from one plate into a test plate. We do this using the acoustics. It's contactless, very high quality, very accurate and very precise. And one of our scientists, Ian Sinclair, who works in our compound management group, looked at that technology and recognised that there's a lot of similarities between the way the acoustic droplets are generated and some of the Taylor cone, for example, that's generated in an electrospray mass spectrometry source. So really we then started to think about how we could take that technology we had familiarity with and maybe adapt it so that we could use it as an interface for loading samples into mass spectrometry. So we've started to look at how we can take our standard biochemical assays and maybe configure them in such a way that we can take advantage of the acoustic mass spec platform. This platform can deliver samples into a mass spectrometer at very high rates. So we're able to sample 3 wells per second. It generates 10,000 data points an hour, which is very much faster than the sort of leading electrospray mass spec platforms that are available today, which probably only process samples at sort of 384 per hour. So we're using this, we've taken actually a project that came to us through our Open Innovation Portal. So it was a project that was recommended to us by a collaborator from Washington State. We've taken that target and we've been able to use the acoustic mass spec to screen 300,000 samples against that target. We've identified some interesting chemistry, and we've been able to take that chemistry and really validate it and demonstrate that they are good quality hits using the acoustic platform in a way that we wouldn't have been able to do with any other technology.